Thank you for that introduction, and it's great to be back here at SciCon. I was here last time talking about how evolution has left us with some rather unfortunate glitches and quirks in our body, and I study the evolution of the human genome, so most of my time is spent looking backwards at evolution's effect on us. So it's really refreshing today to be able to look forward and think about how we're going to take charge of our bodies and our minds and our genomes with science and technology and sort of fix some of those glitches that evolution left us with. And I want to begin by reminding everyone that this is actually a long tradition of our species using technology to enhance our bodies and minds. I want to introduce you to what is definitely the most important, the most crucial invention that humans have ever invented, the most important piece of technology in the history of our species. And that is an Oldowan stone chopper, a little over 2 million years old. And I say this is the most important piece of technology in the history of our species because it set us on an entirely new path. With the invention of stone tools, we began to solve our challenges with our minds and our tools and technology rather than our bodies. We met our ecological challenges with our brains and our ingenuity, our sociality, our knowledge, our wisdom, and of course our tools. And that really does put us in a whole different category. The reason why, and this is why we are what we are today, is because it puts evolution in warp drive. Why does it put evolution in warp drive? Because you don't have to wait around for mutations randomly to come up with the fixes to whatever your problems are. You can solve your problems yourself. Just one little tiny example of how powerful this is. Humans are the only mammal that can live in any climate in the world, from the coldest tundra to the driest desert, the wettest rainforest, the grasslands, everywhere. Why? Well, because we put on clothes, we build houses, we solve all of those problems, not with our bodies, but with our minds, our cleverness, our ingenuity. And we're going to keep doing that. We're going to really, this is a very exciting time to be talking about technology and its enhancement on our bodies because we're on the cusp of doing some incredible things. And if anyone thinks that I'm here to talk about, like, are we all going to be cyborgs and that kind of stuff, I want to remind you that this is a science conference, not a science fiction conference. But maybe that talk will be in 10 years from now. We'll see. But I'm going to talk about the near term. Another technology that really changed everything that I think gives us an example is something as simple as fire. Now, fire did a number of things for us, but the one thing that I think proves the point better than anything else is that it changed what we can eat. Right? There's a whole bunch of foods that really were totally unpalatable to us and poor sources of nutrition unless we cook them. Like, have you ever tried to eat a raw piece of meat, a big chunk of meat? Like, you'll spend hours chewing that, and you won't get much out of it. But with this, you can chew a piece of steak in a few minutes. So it changed everything, because what it did was it offloaded the work of our stomach to the fire. The fire does the work for us. It pre-digests our food. And that's what technology does for us. It offloads the work. It does the work for us so we don't have to rely on our bodies, which is, by the way, why our bodies are so crappy. Because when you take the selective pressure off of your body, it's allowed to be as poorly designed as ours is. And you can read all about that in my book, which is available in the bookstore. And find stores near you. Okay, I mean, just more examples of how we've used our ingenuity. If you look at all of these foods, you probably won't recognize these, and even if you do, you wouldn't want to eat them. But what did humans do with each one of these fruits and vegetables? Is we changed them. So this is another piece of technology. These foods are all rich and nutritious. They have lots of calories. They're good for us, mostly if you don't overdo it. And that's just us using our brains rather than our bodies. If the food doesn't suit us, or the tools don't suit us, we create them, and we solve our problems this way. We've even engineered other living things to be our friends. Of course, when you see, this is Bruno, by the way, when you see Bruno using me as his pillow, as he frequently does, you start to wonder who domesticated whom. So what's your favorite human enhancement? Think about bandages, eyeglasses, composite tools we have here. All of these things that allow us to do things with our bodies that we could never do without. So let's talk about what we're up to now. We're now taking charge of our immune system. We're gonna be curing diseases. We're gonna fix all those errors in our genome. And along the way, we're gonna give ourselves enhanced functionality. And most importantly, Most importantly, we're going to be able to finally offer solutions to a whole bunch of illnesses that we have been completely unable to treat until now. And that's the real power of this. And there's a lot of people who are very worried about the future of genetic engineering, and with good reason. But I want to make sure that we don't throw the baby out with the bathwater, because there are a lot of people out there who could really benefit from the tools that we're developing right now. And I'll give you some examples of those. Let's begin with our immune system. Once again, this is nothing new. We've actually been hacking our immune system for hundreds of years. Even before vaccines as we know them today were invented, we've been intentionally exposing ourselves to cowpox in order to make us resistant to smallpox. I don't know if you guys know that, but humans were doing that for hundreds of years. So we've known that we can actually trick our immune system into creating immune response to a pathogen that it hasn't even seen yet. Such the second time that we see that pathogen or that antigen, our immune response is so much stronger, so much so that we don't even usually get symptoms. So we've been hacking our immune system already, but of course this year, or the past couple of years, we've been able to take this once again into warp drive with the development of mRNA vaccines, right? So they're all in the news right now. The first major product for mRNA vaccines was the COVID vaccine. Well, if you take a second, if you didn't know this already, what is an mRNA vaccine? Well, rather than giving you a little bit of a virus or some proteins from the virus or a dead version of the virus, we actually give you, your body, some genetic instructions, and then your own cells produce the antigen that you're going to raise an immune response to. So we've actually hacked your own body to become its own vaccine fabrication machine. Right? And that's why it works so much better. You get such a better response because your muscle cells will put out that antigen repeatedly, and you'll mount this nice, strong immune response. And as we can see, the effect on COVID infection was remarkable. These mRNA vaccines worked so much better and so much more safely than we even hoped they would. Well, now that we have this technology deployed, mRNA vaccines, we can start to use them in other creative ways. There is now being developed a vaccine, yes, I said a vaccine, for multiple sclerosis. And this is, thank you, I didn't develop it, but thank you. There's now a vaccine for multiple sclerosis in trials. And look at these data from mice. So these are mice, this is a mouse model of multiple sclerosis. And on this axis, what you can see is basically a measure of neurodegeneration, of how well their neurons talk to their muscles. And a lower score is of course better in this case, so it's less degeneration. And you can see that with a single treatment, of an mRNA vaccine to one of the antigens which is naturally occurring in multiple sclerosis, and you can see enormous protection for these mice. So this is not in human trials yet, but it's on its way, and I'll show you where we are with that. In another example, so these are patients with melanoma that has metastasized to the lungs and was inoperable. So these patients are terminal. At this point in time, at the time the study was done, no hope for surgery. And if you can see, this is a chemotherapeutic agent, and you can see the size of the tumor. It continues to grow in the face of chemotherapy. When a patient's cancer stops responding to chemotherapy, that's generally when you start making your final arrangements. However, with this fortunate patient, an mRNA vaccine for 4 antigens that are naturally occurring in melanoma, they were able to cause regression of this metastasis. At the time that this paper was published, this patient is still alive. So this is a patient who's been cured of metastatic melanoma, at least temporarily. And remember, we're just getting started with that, right? We're just getting started. Okay, well, hacking the immune system by triggering it with mRNA is one thing. What if we can re-engineer the T cells themselves? So these are killer T cells. We take them out of your body, re-engineer them, put them back in to go after whatever it is that we've targeted them for. Okay? So this is a patient, and look how old this is. This is now 11 years old. So this is a patient that had bone cancer and then was treated with T cells that had been reprogrammed for antigens that were specifically found in their bone cancer. This is personalized medicine in this case. And you can see complete regression of the tumor cell. And I want to show you a video of how this works. So this was a paper of the biotechnology of this, and these green cells here are the target cells. So these are the cells that the T cells are trained to destroy. And they're trained to destroy them because they have the green fluorescent protein. You may have heard of that one. It glows when you shine it with UV light. It grows green. We get it from jellyfish. So we've engineered these cells to grow green, and we're targeting T cells that from the patient. We take them out of the patient, targeting them to find the green fluorescent protein and then go in. And just watch what happens. I'll show this video twice. So there's the target, and here's this little cell that's found it, and boom. I'm going to watch it again. Here's this tiny little killer T cell. He's found his target, and boom. So imagine if that was a cancer cell, like we've shown in here, or any other cell that you want to get rid of. We now have the ability to engineer your own cells, give them back to you to go do their work. Well, you all probably have heard of CRISPR. Is there anybody in this room who hasn't heard of CRISPR, right, by now? But we now have the ability to edit gene sequences in a living human at will, pretty much. And the reason why this becomes super important is there are a whole bunch of diseases that are actually caused by a single gene and affect very limited tissues. Now, obviously, fixing every cell in your body with gene editing technology, we're nowhere near being able to do that. And I'm not even sure we want to go down that path, right? But there are a lot of diseases out there that are simple, monogenic diseases for which we have almost no hope. Cystic fibrosis, sickle cell anemia. You guys have heard of many of these, and these are, in many cases, debilitating conditions. And they're called rare. Yes, they are rare diseases, but in the aggregate, if you look at all the people who have any kind of inborn genetic disease, it's over 200 million people. So this is not a small problem. Gene therapy has been elusive, and the promise of gene therapy from the 1990s has really not lived up, but CRISPR changes everything. It allows us to do it much more safely. And in fact, its successor technology of base editing is even safer than CRISPR because it doesn't cause double-strand breaks and much less likely to have off-target effects. I'm going to show you, this is just how CRISPR works for anybody who's interested. Just use simple sequence swapping, and we really can edit gene sequences at will. And we're starting to do this now, not just in mouse models, but in people. So I want to show you a study of a condition called transthyretin amyloidosis. So in this condition, patients from their liver make too much of a protein that isn't folded correctly. And when proteins aren't folded correctly, they're dangerous. From the liver, they enter the bloodstream, and they usually accumulate in the heart, but this is a debilitating and lethal condition. So if you have transthyretin amyloidosis, the average time of survival is about 10 years. Well, we've now, and what's fortunate about this particular condition when it comes to gene editing is that you don't actually need the gene that makes this protein at all. We can just simply turn the gene off. And that's exactly what we've now done, is we've engineered a CRISPR technology that simply turns the gene for transthyretin off in the liver of these patients. And you can see, here's a patient the control patient, and how much transthyretin was found at 2 different doses, and you can see the large doses work here to have almost undetectable levels of transthyretin going out a full year with a single treatment. This person went to the hospital and went home, and a year later no longer has the disease. So we're treating amyloidosis now. There's another opportunity that we have. So there's 2 genetic diseases, one beta thalassemia, another one sickle cell anemia, that are both caused by faulty hemoglobin, okay? In beta thalassemia, you basically don't have the beta chains. For those of you who don't know, hemoglobin is made of 4 pieces, 2 alpha chains and 2 beta chains. In beta thalassemia, you basically don't have the beta chains. You need constant transfusions. It is also a lethal condition without very aggressive medical treatment. Sickle cell disease, the severity of it varies from patient to patient, but it's also it's alpha chains that are affected with a mutation, and sickle cell crisis, transfusions. There's a lot of time spent in hospital, and it is also generally often lethal. Well, there's a little trick that would work for both of these diseases. We have another kind of hemoglobin called fetal hemoglobin. When you were an embryo, when you were a fetus, you had a different kind of hemoglobin in your red blood cells that is more aggressive at binding oxygen. And that makes sense for a fetus to have to steal away oxygen from the mother. So in patients with these, they have a problem with either the alpha chains or the beta chains, but presumably their fetal hemoglobin is fine. Gamma, we'll call it. Why don't we just flip that gene back on? It turns off naturally at around the birth. If we flip this gene back on, it could replace the faulty alpha and beta chains, and they could have normal red blood cells. That's the idea. Let's see if it works. So here's a patient with a single infusion of the CRISPR gene in order to turn on, so the gene editing turns this gene back on, and we can see within a couple of months, almost all of their red blood cells are now expressing fetal hemoglobin and contain fetal hemoglobin. What about the clinical outcome? So this is a patient with beta thalassemia. Each one of these dots here represents a transfusion. And if you look at the time screen, in the 2.4 years before they were identified, this is how many transfusions this patient needed. And then during the screening and preparation for the infusion, the transfusion continue with a single treatment with the CRISPR gene to turn on fetal hemoglobin. This patient, almost 2 years later, has not needed a single transfusion. This patient is now cured of beta thalassemia. So here's the case with sickle cell anemia. We have a patient with constant need for sickle cell crisis, hospitalization, blood transfusions, and you can see with one single infusion, we've been able to convert this patient's red blood cells to fetal hemoglobin, and that's what this patient now carries around. Let's look at the clinical outcomes for this patient. Each one of these red dots represents a sickle cell crisis event, a hospitalization where this patient had to go into the hospital for an extended amount of time. And those of you who don't know, it's excruciating, sickle cell crisis is, because you have misfolded red blood cells that essentially get stuck in all the tiniest vessels all throughout your body, and it's awful. It's excruciating pain. Each one of the black dots represents a transfusion, which is necessary for these patients to stay alive. With a single infusion of the CRISPR gene to turn on fetal hemoglobin, this patient has not had a single crisis event and has not required a single transfusion. This patient has been cured of sickle cell anemia. And her name is Victoria Gray. And I think it's important that we remember the babies in the bathwater here. So before we get too upset and scared of gene editing, we have to remember the real people who are now walking around alive and disease-free because of this gene editing technology. So cystic fibrosis is next. Let's see, what else can we do? I told you that we're working on monogenic diseases. So cystic fibrosis affects very few tissues in the body. That's been an advantage. We've already taken human tissue in the lab and been able to cure it of cystic fibrosis. Putting in patients is next. Here's some of the other diseases that we're going to be working on here. Glaucoma, muscular dystrophy, Parkinson's disease, all kinds of cancers. The bottom line is gene editing technologies offers enormous promise, and we're on the way towards curing diseases that we had almost nothing to offer those patients before. Now, even though you're all science enthusiasts, most science enthusiasts also do like science fiction. So let's go there for just a little bit. Why not? What else can we do with this technology? What's possible? Well, sometimes affecting a single gene in a single tissue does have incredible outcomes. And what if you wanted superhuman strength? Could we engineer humans to have superhuman strength? It sounds like a silly goal to have, but there's a lot of different jobs and careers and things out there where people would benefit from that. Well, fixing a single gene, would that ever just make your muscles all big? Well, yes, actually, there's a gene called myostatin, and natural spontaneous mutants of myostatin have been found and discovered. Here's one. A single gene that's been mutated, basically eliminated myostatin, and you don't prevent that skeletal muscle growth. Yeah, but okay, fine, a cow, but could you ever do this in a human? Spontaneous mutants of myostatin have been found in other species as well, including humans. So if it can happen spontaneously with a single gene mutation, then it is something that we could do with CRISPR. I'm not saying we should, but we could. You asked. What about endurance? Well, once again, if we look at the spontaneous mutants that we've already found, there's a cross-country skier whose name I can't pronounce, but he's from Scandinavia, who was a spontaneous mutant in his erythropoietin gene. Erythropoietin is the hormone responsible for red blood cell production. It's, by the way, one of the drugs that performance-enhancing athletes, it's doping. If you use EPO and inject it on yourself, you'll boost your red blood cell count, and that's considered doping. But you can also just train at high altitude, and you will naturally produce, which isn't illegal by sporting authorities. Anyway, this man had a natural mutation, and he had a 25% boost in his red blood cell production, which gave him superhuman endurance, and he won all the medals, etc. What about unbreakable bones? Wouldn't that be nice to have bones that don't break? Well, it turns out 2 different mutations have been found that lead to incredibly hard bones with, in both cases, no other side effects, which is rare. Usually you screw around with one gene, you have these crazy effects that you weren't expecting. But spontaneous mutants of people who have incredibly durable bones have already been found. What about superhuman vision? Once again, there are some people who walk among us who have much better color discrimination than we have because they have 4 color detecting cones instead of 3. We have 3 color detectors in our retinas, and they cover different regions of the electromagnetic spectrum. But there are some spontaneous mutants that have a 4th one, and so they would experience a richer, more vibrant color scheme than the rest of us. And that could also be put to good use, as you can imagine. What about a superhuman sense of smell? Do you guys know that there are mutants that have this incredibly sensitive sense of smell? They usually get hired by the FBI and the CIA because there's like evidence that, ooh, this letter was once in Cleveland or something. They can do incredible things with this sense of smell. And they are mutants of the gene called CAL1, and the condition's called hyperosmia. And it can even be a side effect sometimes of other diseases. But a mutation in CAL1 leads to a superhuman sense of smell, which once again could be helpful. What about short sleepers? Well, spontaneous mutants of a gene called DEC2 has been found running in families, and in fact, my PhD advisor he is from one of these families. He needs 3 to 4 hours of sleep every night, and that's enough. He never gets more, never wants more. So he has an additional 3 or 4 hours than the rest of us every single day of his life, and you can imagine how helpful that is. And by the way, he's 83, and he still runs 2.5 miles every day. He's down from a larger amount, but he's in great shape, so he's not suffering any ill effects of his lack of sleep. He hasn't even retired yet. Okay, now I know everyone's getting a little nervous. I can read the energy in the room about all this hacking of the human body and designer babies. Let me tell you why I'm not worried about it in the short term. We actually know very, very little about how to hack the human body in whatever way we want. So what will we do with designer babies? Make them more attractive, make them more intelligent, make them taller? Well, let's look at height. Do you guys know how many genes are involved in human height? Anybody have a guess? 36. 100. So, 800 genes are known to affect human height, and that only accounts for 1/3 of the heritability that we know human height has. So there's another 1,600 that we don't even know what they are. And all of those genes do other things in the body. So we are nowhere close to being able to tweak your genetics to make you taller, let alone more attractive or intelligent or any of these things which are much more complicated genetically. So I am not worried about this in the short term, and I'm certainly not willing to sacrifice patients like Victoria Gray because some people are worried about designer babies. That's why I say that. Okay, so the last topic I want to talk about is human-machine interfaces or brain-computer interfaces. This technology has also recently undergone leaps and bounds of development. And this is because our ability to read the brain waves has increased, and our ability to deliver the instructions from those brain waves to human tissues. So this really isn't that complicated because we don't have to understand what all the waves are, where they come from, and what they do. We just have to discover them. So we put them in someone's brain and we tell them to do something, or if they're paralyzed, as we'll see, to think about doing something. And we just watch what the brain does, code that, remember it, and say, okay, the next time the machine sees that, do something. And it leads to some pretty phenomenal results. This needs a lot more development, but remember, this is the Kitty Hawk flyer, and how long Kitty Hawk was the first airplane, the heavier than air airplane. Anybody doesn't know. So the Kitty Hawk flyer, to how many? It was just a couple of decades before we had, warplanes. So imagine this is the Kitty Hawk flyer of these human-machine interfaces, and let me just show you where we are with this.
Depending on what electrode they stimulate on, there are different sensations I can feel across different points on my hand. So sometimes it's pressure, sometimes it's just a tingle, warmth. The first time it was really cool. It's kind of like, did that really just happen? now it's just second nature. When I was 18, I was in a car accident and broke my neck. So now I'm a C5 quadriplegic. When I was in the hospital in Pittsburgh, they put me on a research registry. So they gave me a call and were like, hey, we think you might be interested in this. I was like, yeah, robots are cool, so.
Yeah, I remember just now.
Nathan first volunteered for surgery, so he had to go in and have neurosurgery and spend a week in the hospital. After he recovered from the neurosurgery, he's pretty much been coming here 3 times a week. He spends 4 or 5 hours here each time he comes.
He has been implanted with microelectrode arrays in his motor and somatosensory cortex. You can see that we're recording neural signals from the motor cortex, and we are decoding these signals so that you can control a robotic arm.
Okay. I thought in the past it would light up blue. Okay.
Essentially, he thinks about moving his arm, moving his hand, and so different neurons will be more or less active as he thinks about moving in different directions. And as we record from a whole population of those neurons, we can essentially decode which direction he's thinking about, whether he's thinking about opening or closing the hand, moving his wrist, and turn that into a control signal for the robot.
He wants to help other people. Everybody in the room who's testing Nathan, everybody who's been involved in this project has as a goal to cure paralysis in one form or another. From the very beginning, he said, you know, if no one does this, then the work won't advance.
I hope that when people have a traumatic injury, that they don't feel like their lives are ruined, that one day there will be cures for things like spinal cord injury. If you have lost a limb, you can get one that's maybe even better than the one you had before.
So that's really great. He's able to move a robotic arm by just thinking about it. And he's a paraplegic, so the immediate use of it is obvious, but imagine what we could all do. We could control all kinds of devices with our brains. You don't have to be paraplegic for this to work. But what about restoring function to your own body? I mean, that would really, moving a robotic arm is fine, but that's not very practical. You can't imagine going to the grocery store, with a robot, and and it wouldn't be any easier than the life they have now. Another 10 or 12 years, imagine where this technology will be. So we're offering a new life to patients who have lost functionality. And this is also being applied to the language centers of the brain. And it doesn't work very quickly, but we've had patients who have had their speech essentially restored using a computer and thinking about certain words. Again, you had to train the machine because it doesn't know what "I am very good" looks like in your brain, but you do. So you can think about "I am very good," and it remembers that. And so once you train the machine on each word, we can recall it. So the last topic that we're working on hacking is mortality, aging itself. That's the ultimate goal of our wish to hack our bodies. And here's why I'm somewhat hopeful about this, is that, I don't know if you guys know this, but we age on purpose. Aging is a programmed event. We are all designed to get old and die. Why? Because evolution doesn't want to keep you around forever, right? You need to get out of the way and make room for the next generation, because otherwise we wouldn't be adaptable. That's how adaptation and evolution happens, is by constantly bringing in new genes and new combinations of genes. So if we can figure out the program and slow it or stall it, we might be able to make big jumps in our ability to stop aging. That research is now ongoing. And in fact, we've been able to undo as much of the aging as we think is theoretically possible, and we have been able to predict that the upper limit on human lifespan before we really just can't keep going anymore is between 120 and 150 years. So I would say that within a generation, most of the population will be living between 120 and 150 years if research like this continues. So I think the future is bright, and the future is now, and there's a lot of things to be very excited about. Thank you very much.